Hello, good afternoon, everyone.
Good afternoon, Bu Elsa.
Oh, Bu Josephine,
Hai, apa
how are
kabar?
you? Okay, I think all of the presenters already here. So we have the judge. Good afternoon, Dr. Henry.
Good afternoon, Dr. Elsa.
Yeah, and good afternoon also, Dr. Astri.
Yeah, good afternoon, Dr. Elsa. Happy to be here.
Okay. Okay, so Dr. Asri and Dr. Henry, the presenters are all already here in this breakout room too. So should we start?
Yeah.
Yes, please.
Okay. So good afternoon and welcome for all of you. This is the sessions, oral presentation sessions with topic malaria and One Health. So here, actually, we have four presenters and three judges. However, one presenter, Bapak Ima Dewi Narta, yeah, uh, we just informed by the organizers that Bapak Made cannot join today because of something that's very urgent. So finally here we only have three presenters. I would like to introduce to you the judge for these sessions is Dr. Henry Surendra, MPH, PhD, IFRS, and also Dr. Asri Ferdiana, MPH. And there are three speakers here. First, Miss Wihda Aisarul Azmi, Miss Mesyalie Lorenza, and also Miss Andita Fitri Mutiara Rizki. Yeah. So this is the rule for the sessions. So first, the speaker will present their presentations for 7 minutes. And after that, there will be questions from the judges, yeah, for 5 minutes. So please, the judges, please ready to take the score for the presentations. So here I would like to say hello first to Wihda. Wihda, are you here?
Yes, Miss Elsa, I'm here.
Hi, Wihda. Are you ready for this presentation?
Yeah, I'm ready.
Okay. So without any further delay, so let's start with the first presenters, Wihda Aisarul Azmi. Okay, you ready, Wihda? Please, the 7 minutes is yours.
Okay, first, thank you. First of all, I would like to share my screen. I'm sorry, Miss. The host has disabled participants' screen sharing, so
Okay, you cannot share your screen.
you can
So
share. Yeah.
okay, let me. So Wihda, can you just try to share the screen?
I still can't share it, Miss.
One Wihda. Okay. Please, Mbak Anggit. You hear, Mbak?
Yes, Bu Elsa.
Ya,
Sunscreen sudah bisa, Bu.
oke.
Okay.
So please try again, Winda.
Okay. Okay, thank you very much. Good afternoon to all of the participants in today's conference. I am Wihda Aisarul Azmi from Master Program in Biomedical Sciences, Universitas Indonesia. Thank you for having me here to present my current study, Development of Rodent Malaria Parasite Model Resistant to Artemisinin. Malaria is one of the infectious diseases that caused human mortality and morbidity and still a huge threat to public health. The World Health Organization has been conducting global technical strategy for malaria in order to achieve malaria elimination in 2030, especially in the 85 endemic countries. The World Malaria Report 2021 has reported that there is an increase in the global estimated malaria cases and deaths from the 2019 to 2020. And this happened because of the COVID-19 disruption and mostly the parasites' resistance to several antimalarial drugs. The antimalarial drugs resistance has been one of the major hindrances in malaria control. The first resistant case was found in the 1950s when the chloroquine-resistant parasites occurred, followed by the parasite resistance to other antimalarial drugs. The Artemisinin combination therapy, which is used to combat that antimalarial drug resistance, has been found that there is also an indication of the resistance of the parasite to this drug. With no alternative treatment available, the new antimalarial drug development is urgently needed, since we also know that in population we can conclude that there is already so many resistant and sensitive parasites spreading in the population. And the new candidate of antimalarial drug should be able to clear both of the resistant and sensitive parasites. Thus, to screen this candidate antimalarial drugs, we need the parasite model resistant to several antimalarial drugs, and our laboratory has been successfully developed parasite resistant to atovaquone and pyrimethamine, and with the same method within host selection, we would like to develop new parasite resistant to artemisinin. To develop stable artemisinin resistant parasites, we use the called repeated incomplete treatment method, where the parasite will be exposed to an inadequate dose of artemisinin to clear the parasite without finishing the treatment process, so that we could get the resistance of the parasite to mimic the natural infection in the population. We use biopsy mice infected with Plasmodium berghei that caused to 3 different regimens of RICT treatment. The RICT cycle would be started when the infected biopsy mice percentage of parastemia reach 2% to 5%. The drug administration will be done in 24 hours until the percentage of the parastemia fall to 0.5% or less, and let the parasite to recover, and the percentage of the parastemia increased again to 2% or more. And after that, the drug treatment would be continued, and this is the start of a new cycle. These cycles would be repeated until we can find the possible phenotypic resistance. This is the result in this study. This graph shows the features of the RICT experimental system in the first group, where we use the dose of artemisinin 10 milligrams per kilogram body weight treated every 24 hours. We can see that from these mice, it went on 2 days of treatment in cycle 1, followed by 3 days on recovery period, and 10 days of treatment in cycle 2. The percentage of parastemia increased dramatically on the day 9 and steadily increased until the end of the cycle 2. And this is the summary of the treatment and recovery times observed in this group. We used 12 mice, and the average of times that needed to treat in the cycle 1 is for 3 days, and the average time for recovery cycle is 3 days too, and the mice needs 6 days in average to finish the cycle 2. This is the second regimen where we treat the mice with 20 milligrams per kilogram body weight of artemisinin every 24 hours. And from this graph, we could see that the mice only last for 1 day in treatment in cycle 1, and followed by 3 days of recovery period and 10 days of treatment in cycle 2. The percentage of the parastemia also increased dramatically on the day 8 and relatively stable until the day 14 of the RICT. The average days of treatment needed for cycle 1 in this group is 2 days, and the recovery period is 3 days. And for the treatment in cycle 2 is 5 days. This is the third one, where we treat the infected mice with 10 milligrams per kilogram body weight of artemisinin every 12 hours. We can see that the slope in the graph and this regimen is much different with the other two regimens. We could see that percentage of the parastemia in this regimen is much lower. Yeah, in this mice, they spend 3 days in treatment in the cycle 1, 3 days for recovery period, and 3 days for the treatment periods in the cycle 2. And for your information, for this third group, it is still ongoing experiment, so we can still update this data since the mice right now still in the middle of the cycle 2. And this is the summary of the treatment and recovery times needed. The average days they need to spend the cycle 1 treatment in cycle 1 is 4 days, followed by 4 days for recovery period, and in average 2 days of treatment in cycle 2, and it's still continuing right now. And the conclusion of the studies is the regimen of artemisinin dose 10 milligrams per kilogram body weight treated every 12 hours seems to give the most promising result in developing a stable artemisinin-resistant parasite. And for the ongoing experiments, we would like to do the molecular analysis, the whole genome sequencing, to confirm the possible resistance of parasites that could have been obtained. I think that's all I can present today. Thank you very much for your attention.
Thank you, Wihda. Applause for Wihda. So exactly 7 minutes, clear and nice presentation. So I would like to invite the first judge, Dr. Astri, please.
Yeah, thanks very much, Dr. Elsa, and thank you, Wihda, for the nice presentation. Actually, I'm not really an animal scientist, so if you can explain to us what is the implication of your main findings to the regimen of artemisinin for human? Is there any implication that we can learn?
Okay, thank you for the question, Miss. We tried to develop the artemisinin-resistant parasites here in rodent malaria. Is for the model. We use the method repeated incomplete treatment, where we develop based on the natural and human infection. So we hope that with this resistant artemisinin parasites that developed through this RICT method, we could use this parasite to analyze the mechanisms of the resistance in the artemisinin-resistant parasites, and we could also use these resistant parasites on the new antimalarial drug screening method, so that we could find the new drug candidates that could also clear both not only the wild type parasites but also the resistant parasites, and for this one specifically for artemisinin-resistant parasites.
Yeah, thank you. Do you have any data on to what extent the resistance to artemisinin has been found on human?
The artemisinin resistant that has been found in human. If I'm not mistaken, it's already found since the 2008 in Cambodia, and not in the recent years there is also the findings in Africa too. So since the artemisinin is one of the first line antimalarial drug that used in ACT, I think it's really important for us to understand more about the resistance of this drug, and this study could help that.
Any evidence of artemisinin resistance in Indonesia that you know of?
In Indonesia, the latest data I
evidence.
found in West Sumba of Indonesia, it's indicated that it is also artemisinin resistant.
Okay. That's all for now, Dr. Elsa. Thank you.
Thank you, Dr. Asri. So I invite the next judge, please, Mr. Dr. Henry.
Thank you, Dr. Elsa. I think Dr. Asri already took the question actually, but congratulations to Wihda for the nice presentation. Maybe only one question from me, because I saw in your conclusion you said that your conclusion seems to be like not really conclusive to me, because you said it in not a firm way. So do you think that in your statistical analysis that you can perform a more robust analysis, so that you can then firmly say that it is significantly that the finding that you found is really significant? Because I saw the graph that you plot is basically like descriptive graph. Is there a way to do it? Thank you.
Okay, thank you for the question, sir. Actually, from this data, I still couldn't do the statistical analysis. So, since my objective is to develop new artemisinin resistant parasites, like I did the selection from the wild type parasites to get the artemisinin resistant parasites. So this is still like a preliminary study, like how is the features or how the pattern of RICT treatment that I did to develop this artemisinin resistant ghost. And if I already based on this data, like I already see that the pattern of the RICT cycle in the third regime is more clear between the treatment periods and the recovery periods rather than the other regimes. And the other reason why I said that the third regimen is the most promising one is because in the first and the second regimens, we could see that there is a dramatic increase of percentage of parastemia on the day 9 and the day 8 in average to all of the mice. And the amount of the percentage parastemia is too high. Like it could disrupt the RICT process, since if the parastemia is too high, it could also affect the drug effectiveness in clearing the parasites. So I hope with the current data in the third regime, I could get the resistant parasites like my objections.
Yeah, okay. I think that's all from me, Dr. Elsa. Back to you.
Okay. Okay, thank you, Dr. Henry. My question, Wihda, looking at the result of the two therapeutic group, I see that the 10 milligram per kilogram body weight is so promising result. Is it right? And inducing the stable artemisinin resistant parasite. So, do you think that for somehow the resistant artemisinin the parasite that resistant artemisinin you develop can turn to be susceptible again for after several cycles? Is it possible, Wihda?
Thank you for the question. The objection is to find the stable artemisinin resistant parasites, so that if we grow these isolated parasites from this experiment into a new boxing mice, it still have its resistance. It still have its resistance, so that if we treat the these parasites again with artemisinin, the drug would not be effective to clear the parasite. And for the 10 milligrams per kilograms body weight treated for 12 hours, I said that it could be the best regime to develop the artemisinin resistance, because just like I said before, that we can see the clear pattern, and we can see the clear pattern. And sorry, for the what can I say? If the artemisinin resistant parasites have a stable resistance, it could stay for a long time, even though we already cryopreserve the parasites in a long time. So we hope that even if we grow this again, the resistance is still there.
Okay. And your suggestion, Wihda, you suggest that the next step in this study is to investigate the predicted gen target corresponding to artemisinin resistant. Would you like to explore more specific about this gene target? Is it like, let's say, Cal13 in Plasmodium falciparum, or what?
Yeah, I would like to elaborate more on the possible mutations that happen in this resistant parasite, since the reason why we use RICT is the RICT was developed to mimic the natural infection in human, and as the experiments, as the experience in our laboratory, that we already have developed an autophacon resistant parasites. It turns out that the autophacon resistant parasites we get at that time was dose dependent, and it could be different if we use different. It could give different mutation degree if we use different method to develop it. So we use the RICT to mimic the natural infection that happens in humans, so that we hope that the mutations that occurred in this resistant parasite model, could represent what that happened in human.
Okay, thank you, Wihda, for your answers. Thank you very much. And now we will move to the next presenter. Thank you, Wihda.
Thank you very much.
Okay, everyone. Yeah, I would like to introduce you to the next presenters. We have Mesyalie Lorenza. Hello, Mesyalie. Mesyalie or Lorenza?
You can call me Mesya.
Mesya, okay, Mesya. So, Mesya, you will present your works on systematic review. Is it right? Systematic review Plasmodium falciparum transferases as potential targets. Is that right?
Yes,
Okay.
that's right.
Okay. So, Mesya, the Zoom is yours for 7 minutes, please.
Okay, I will share my screen. Okay, is my screen visible?
Yeah.
Okay. Greetings to the Honorable Committee, judges, and my fellow participants. My name is Mesyalie Lorenza, coming from the Department of Biomedicine at Indonesia International Institute for Life Sciences. And first of all, I would like to thank you for providing me the time to present our systematic findings on the Plasmodium falciparum transferases as potential anti-malarial targets. So I believe that most of us here have been familiarized with malaria, which is an infectious disease caused by the Plasmodium falciparum and caused by the Plasmodium species. And among the 5 Plasmodium species, the Plasmodium falciparum is the most life-threatening species because it can lead to rapid progression to severe infection. And unfortunately, this species also accounts for more than 90% of the malaria-associated deaths. So although this parasite has been identified and researched since 1980s, the elimination of this parasite remains very challenging. And one of the hurdles to eliminate Plasmodium is because of the spread of multi-drug resistant Plasmodium falciparum strains. So Plasmodium has an extensive ability to undergo gene mutations that will alter the structure of the targeted enzymes. So as we can see here, they can alter the active site of the enzymes so that drugs that previously designed to target this structure does not interact with the enzymes anymore. Or otherwise, Plasmodium also can express efflux transporters, so the drugs will be omitted out of the parasite cells. That's why we need a new anti-malarial targets. And in this presentation, I would like to bring Plasmodium falciparum transferases into the discussion. So generally, transferases are a class of proteins that help the transfers of a functional group from one molecule to another molecules. And in Plasmodium falciparum, it presents abundantly and also involved in various biological processes such as gene expression, protein localization, cell cycle progression, and antigenic variation. But unfortunately, this class of proteins is less studied and understood for anti-malarial targets. That's why we want to summarize all information regarding the potential of Plasmodium falciparum transferases to be targeted for malarial drugs. And that's to achieve that goal, we did systematic review. And here, the systematic review was done by 3 reviewers, and we managed to collect more than 1,000 articles, but of these 1,000 articles, only 20 articles that fulfill our study criteria and also eligible for final inclusion. And from these 20 articles, we identified 11 drug-interacting PF transferases that present in either sexual or asexual blood stages. So what means by drug-interacting PF transferases is that the transferases has been tested against drugs. So they showed interaction when treated with drugs. And from these 11 drug-interacting transferases, we can actually categorize them into based on their prominent function, such as the metabolic regulation, epigenetic regulation, and lipid biosynthesis, as well as the protein lipidation. So this is the 11 drug-interacting transferases. 3 proteins are associated for H metabolic regulation and epigenetic regulation, while the other 5 proteins are associated with lipid biosynthesis and protein lipidation. A more detailed description will be covered in the next slide. So yeah, the first class of proteins are associated with metabolic regulation. So we can see here there are NMNAT, ATC, and HMT, which generally they are involved in cellular metabolism to produce energy, and some also produce metabolites that are important for DNA or pyrimidine synthesis, basically our DNA or RNA synthesis. And some drugs that have been tested against this class of proteins are NMNAT inhibitors, mTOR inhibitors. mTOR inhibitors itself is a repositioning drugs. So previously it was used to treat cancers, and we also have pyrazolopyran-based compounds. And the next class of protein is associated with epigenetic regulation. This includes histone lysine methyltransferase, histone acetyltransferase, and histone arginine methyltransferase. So all of these transferases are involved in post-translational modifications of the histone proteins, and they are involved in the regulation or expression of virulence genes or genes associated with the growth or replications of the parasite itself. And some drugs that have been tested against this class of proteins are G9A inhibitors. Again, this is a repositioning drugs that previously used to treat cancers also. We also have ADOX and HSTIB60 inhibitors. And the last class of drugs, we have PMT, PPT, ISPD, NMT, and DHHC PATs, which are associated with lipid biosynthesis and protein lipidation. So all of these 5 transferases are involved in the synthesis of cell membranes, so which means they are very important and essential in the growth of daughter cells of the parasite, and some also important for the transmission and also invasion or interaction with the host cell membranes. And some drugs that have been tested against these types of transferases are amodiaquine, tetrahydroquinoline, phosphamidomycin, and also benzothiophene and 2-bromopalmitate. Some of these drugs are repositioning drugs like phosphamidomycin. We have used this as antibiotics, but researchers also try to use these antibiotics to target parasites. So to conclude my presentations, I would like to emphasize that Plasmodium falciparum transferases are potential targets, potential novel targets for antimalarial drugs, because it is involved in the parasite, in the growth of the parasite, the pathogenicity, as well as the virulence of the parasites. However, most drugs that have been mentioned before and also have been examined by the previous studies exert antiplasmodial effects at micromolar concentration, which is quite high compared to currently available drugs. Therefore, for the recommendation, it would be great if researchers can optimize the structure and the size of existing candidate molecules through in silico approach. And for drugs that have exert potential antiplasmodial effects, it would be great to investigate the pharmacokinetics of the repositioning or candidate drugs. So that is all from my presentation today. Thank you for your attention.
Okay, Mesya, thank you and congratulations for your systematic review result. Yeah. So now I would like to invite Dr. Henry for the first question, please, Dr. Henry.
Thank you, Dr. Elsa. Thank you very much, Messi, for your nice presentation on the topic of the systematic review study. I think one important question will be, I don't want to criticize the methods because, you know, some people will argue that systematic review is not research, but some people will argue it is research. What I wanted to follow up is regarding your recommendation about the next research following up this your conclusion. So if you have money to do more robust research, can you please elaborate your what will you do you know, in regards to your recommendation? Thank you.
Okay, thank you for the question, Mr. Henry. So based on our systematic review, we managed to find several potential proteins compared to other transferases. So there are 4 most potential transferases that has been tested in vitro and in vivo that they exert high selectivity against human cells, so like 1,000-fold selectivity compared to the human cells. So these four most potent proteins fall in the categories of metabolic regulation and also lipid biosynthesis and protein liquidation, such as the NMNAT and also ATC and HMT. This drug, and they also have several drugs tested against these proteins. So if I have the money to do research in the lab, I would like do screening, like hit screening maybe, like to find the potential substance that interacts with the drug, that exert the most potential or the most therapeutic effects that could interact with these proteins and also have safety profiles to be consumed by humans. But before that, I think in silico approach would be beneficial because previous studies have shown that these proteins are exerting anti-Plasmodium effects that are potential. But by using repositioning drugs, they have like their concentration are about micromolar concentration, which is quite high compared to currently available drugs. So I think by using in silico study, we can like polishing the structure of the enzymes so that polishing the structures of the drug so that it will target and interacts stronger with the enzymes.
Thank you. I think that's all from me, Dr. Esa.
Thank you. Next, Ibu Astri, please.
Yeah, thank you, Dr. Elsa, and thank you, the authors, for the presentation. I would like to ask, because you mentioned that you did a systematic review, could you elaborate, because in systematic review, we usually use the PRISMA guideline and also PICO principle. Could you explain each of the PICO, the population intervention and everything, to your, how do you apply this PICO to your systematic review, and why did you use systematic review approach rather than a simple narrative review or scoping review?
Oh, okay, thank you for the question, Bu Astri. So in our systematic review, I don't think PICO does not like really align with our study because we don't really compare the outcomes of.
So I don't think, sorry to interrupt, I think it's the other way around. Your study is not really in line with systematic review principles. Because in systematic review, we usually, systematic review came from clinical medicine. So it used population intervention, comparison, and outcomes. So I don't think how you can apply the systematic review principles into laboratory science review, like what you did. Okay. So I'm just wondering why you call your study as a systematic review rather than a narrative review or scoping review. I think it's more suitable for scoping review.
Okay, we call this systematic review because we gone through the process of systematic review. Like we did identify, we did set up the search terms for the systematic review. We also like extract all of the articles that align with our search terms into.
But you also do that in scoping review. It's not exclusive approach for systematic review, but in any kind of review, including scoping review, you also use that kind of approaches, selecting keywords and selecting articles, abstracts, and then extracting abstracts. It doesn't mean that it's systematic review, because in any kind of review, you would do that anyway.
Yes, but I think that one that makes us different from other review is that we did the eligibility criteria studies, which is also
yeah,
applied.
that's also done in a scoping review. You also make inclusion criteria on which kind of studies that you would include in your scoping review. So I think you and your team should think again, because when you submit it to a journal, I think you should be very clear that is it a scoping review, narrative review, or systematic review? So don't say that the PICO statement or the systematic review principles is not in line with your study. I think you need to decide which study design you need to think again about your study design, whether it fits with the principles of systematic review. It's not the other way around. Okay.
Also, what you mean is that all systematic review will be using the PICO principles?
Most of the time, yes.
Okay.
Yeah, but also it depends on the journal, because some journals can be very strict. I don't know about laboratory science journals, but yeah, as Dr. Henry mentioned in the chat box, people sometimes confuse systematic review and narrative literature review. Sometimes people thought that doing it in a systematic way can be called systematic review, but it's not. Because also in narrative review and scoping review, we also do the same kind of steps. So please, you should check again the principles of systematic review and see if your study fits with their criteria. Otherwise, the journals would not accept it.
Thank you for the insights.
Thank you, Dr. Astri. So, Mesyalie, this is an interesting study to investigate the new drug target, yeah, for Plasmodium falciparum, but maybe the voice that the terminology you choose is not the right one, the not systematic review. Yeah, I'm just curious about the searching term you use in this investigation. What's the search term?
Okay, we applied 3 search terms, which is the Plasmodium falciparum and blood stage transferases and drug study design. So, which means that all transferases, all the blood stage transferases have been tested against drug.
Oh, okay, also, okay. Okay, so is it in the paper you found? Is it all in vitro, or is there any in vivo and ex silico, etc.? Or it mixed?
They are mixed. Some are in vitro, some are in vivo, and some also in silico.
Yeah, I think that will be a little bit hard, yeah, to compare the outcome of the drug target if we have like a different set of different study setting. Yeah. It's just my suggestion. Okay, thank you, Mesyalie.
Thank you.
Yeah. Now, last but not the least, we invite the speaker, Andita, Fitri, Mutiara, Rizki. Hello, Andita.
Thank you, and hello, Ms.
You will present your study, experimental study, right? You have experimental study, yeah, in searching new anti-malaria drug candidate. Okay, and in Andita, yeah, Andita, you have 7 minutes, yeah.
Okay.
To present.
Okay, thank you. First of all, I would like to share my screen. Yes, good afternoon to honorable judges and all participants. My name is Andita Fitri Rizki from Master Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia. Thank you for having me here. In this occasion, I would like to deliver my research presentation titled Effect Extract Mangrove Plant Sonneratia alba against Plasmodium berghei sensitive and mutant atophaquan. Sorry. Yes. Malaria is one of infectious disease that cause major burden for health aspect in the world, caused by parasite named Plasmodium and transmitted by mosquito named Anopheles. And according to World Health Organization 2021, 38% of malaria cases in the world come from Southeast Asia region. And one of concern faced to control malaria disease is antimalarial drug resistance, and this is required solution by developing new antimalarial drug. And the candidate of new antimalarial drug is expected not only to kill sensitive parasite, but also the resistant one. And the variation of sensitive and resistant parasites is representing the condition in the field. As we know that Indonesia is a country with very high variation of biodiversity, and one of them is mangrove plant, Sonneratia alba, or as well known as berpat. And based on the earlier study, Sonneratia alba has antimalarial activity in Plasmodium berghei sensitive in ex vivo. And also Sonneratia alba extract has activity to inhibit dehydrogenated dehydrogenase or DHODH enzyme that found in mitochondria malaria parasite. And our group has also established the atophaquan resistant malaria parasite model in Plasmodium berghei Y26HS mutation. So the purpose of this study is to know the antimalarial activity of Sonneratia alba plant extract against malaria parasite, Plasmodium berghei sensitive, and atophaquan resistant Y26HS mutation. And the objective of this study is to investigate the efficacy of Sonneratia alba extract in Plasmodium berghei sensitive and atophaquan resistant Y26HS mutation using in vivo method. And the method delivered in this research is antimalaria activity test using in vivo method in rodent model. The rodent model that used in this research is groups of palsy mice 8 to 12 weeks old, and the parasite is the Plasmodium berghei sensitive and Plasmodium berghei atophaquan resistant Y26HS mutation, and the extract itself is Sonneratia alba plant extract methanol. And the treatment method that conducted in this research is by using intraperitoneal injection. And then we divided 6 groups of infected palsy mice, and one of group of palsy mice is consists of 3 mice, and 4 groups of infected palsy mice were treated by Sonneratia alba extract in various concentration, and one of groups is for negative control, which is untreated. And then for the one group is for positive control, they treated with atophaquan therapeutic dose 15 mg per kilogram body weight, and the treatment is conducted for 4 days treatment. And then we monitor the parasitemia level of each mice during 4 days treatment using blood smear. And then this is the growth rate of the parasite Plasmodium berghei sensitive that treated by Sonneratia alba extract in ex vivo. We could see in here that Sonneratia alba extract, the Plasmodium berghei sensitive that untreated control and treated group with the Sonneratia alba in 0 hour before incubation. In this one, only showing in ring and trophozoite stage. And after 24 hours after incubation, the untreated control parasite showing the growth of parasite from ring and trophozoite stage to the schizen stage. Meanwhile, in the parasite Plasmodium berghei sensitive that treated with Sonneratia alba extract in various concentration is not showing any growth of parasite from ring anthropozoite to schizen. And this pattern has similar pattern with the Plasmodium berghei sensitive that treated with atofacon drug as positive control. And this is the graphic of growth curve of Plasmodium berghei atofacon resistant Y26HS mutation that treated by Sonneratia alba in various dose of concentration. We could see that this is one group of positive control that treated using atofacon therapeutic dose 15 mg per kilogram body weight, showing any increase of parastemia level during 4 days treatment. Also, the untreated group and the other group of mice that treated with Sonneratia alba extract in various concentration. And this is for the growth rate of the group of mice parasite that treated with Sonneratia alba extract in various concentration. We also could see that group of mice that treated with atofacon therapeutic dose 15 mg per kilogram body weight is showing the decrease of parastemia level during 4 days treatment. Meanwhile, the untreated group of parasite and the other groups showing the increase of parastemia level. So the conclusion of this research is Sonneratia alba extract inhibits growth of Plasmodium berghei sensitive in ex vivo. Meanwhile, Sonneratia alba plant extract did not show any antimalarial activity in vivo against Plasmodium berghei sensitive and atofacon resistant Y26HS mutation. So this brings me to the end of my presentation. Thank you for your kind attention.
Okay, thank you, Andita. Yeah. So the first question, that's from me. Just clarification, Andita. What part of the mangrove plant that you use for the extract here?
From the extract, we use plant extract from the leaf.
Yeah, I think you should mention it, yeah, because it may be leaf, maybe root, maybe also stem, yeah. So, okay, that's clear now that it's from the leaves. Okay, and Andita, is there any explanation why the extract can inhibit the growth of the Plasmodium berghei sensitive ex vivo, but not an in vivo study?
Okay, thank you for the question, Dr. Elsa. Because for the experiment in ex vivo, we could screen that this Sonneratia alba extract has anti-malaria activity that inhibits, they can inhibits the growth of the parasite in ex vivo. But why not in vivo? Because in vivo is we use living organisms such as mice as the model, and then we should consider the metabolism of the mice itself. So I think they have different results from ex vivo and in vivo experiment from antimalarial testing.
Okay, so for the conclusion, will you continue this experiment or not? If there is the in vivo show unsuccessful.
I think I will continue this experiment, but for the future plan for the experiment, I will redo the experiment because this research requires redo the experiment, especially for the in vivo experiment, and also considering about the biophytochemical of the extract itself, and then probably using another batch of the extract itself.
Okay, thank you, Andita. So please, Dr. Henry, question.
Thank you, Andita, for your presentation. My first question will be about your sample size. I saw you presented that you have 6 different group of mice, but I don't see the description of how many, what is the sample size per group that you have in this study. That's my first question.
Okay, thank you, Dr. Henry, for the questions. For each group of mice, we use 3 mice per each group.
Okay, 3 mice per each group.
Yes.
Okay. I think this also related to to your results data, because one, do you think 3 will give you enough sample size to show the pattern? Because you will need to do like not only, I think in my mind, it's what you need to do is not only do the descriptive statistic, so showing it the trend, but you also can do like perform trend analysis. You can test whether the trend is significantly statistically significant, and also you can then compare not only assessing the trend, but also you can for example quantify the what you call it, the difference between the level of parastemia, if I'm not mistaken, between each group. You know what I mean?
Yes.
Yeah. You want to explain that? Thank you.
Okay. Thank you for the questions. Probably for the next, I will do the more analysis for the parastemia level itself, considering the number of mice that I use per group, and then probably for the next future for the research plan, probably I will use more mice per group, considering to the data for analysis for the parastemia level itself.
Yeah, I think because now you just rely your your conclusion based on those graphs, then you decided because the one is showing like the decline trend, and then you said that this is work, the others not work. But if you see there's different level of parastemia between those other groups, and you can quantify the different, and then see which groups show the most decline in parastemia over time also. Thank you.
Okay, thank you, Dr. Henry.
Okay, thank you, Dr. Henry. Next, Dr. Astri, please.
Yeah, thank you, Dr. Elsa, and thanks for the presenter for the nice presentation. I would like to ask again, what is the implication of your study findings to malaria in human, if there is any, or is it still too far from human studies?
Okay, thank you for the question, Dr. Astri. Okay, the implication of this study is to knowing the efficacy of the Sonneratia alba itself as new candidate of antimalarial drug, considering we have that preliminary data of the ex vivo that we screen, that we have that this Sonneratia alba is have antimalarial activity that inhibits the growth of Plasmodium berghei sensitive parasite. So I think for the future study, this study also have a change to the for the malaria in human, especially because in here we use parasite model in rodent is the basic research, because before we use the human malaria, we should try first in the model rodent. I think.
Yeah. But how prevalent is this Plasmodium berghei in human? Is there an human infection by Plasmodium berghei? And if not, can we imply that if this compound that you found would also be effective for the more prevalent Plasmodium in human, like falciparum or 5X?
Okay, thank you for the question, Dr. Azmi. So this Plasmodium berghei why I use this Plasmodium berghei, because Plasmodium berghei is having a similarity with the malaria, human parasite malaria, like Plasmodium falciparum. And why we use the Plasmodium berghei model to the basic research first, to the develop and discover the new antimalarial drug, because this Plasmodium berghei has a similar, beside of the similarity with the human malaria parasite, this Plasmodium berghei have this cycle of it's for 24 hours, because is promising. I think it's a promising model for antimalarial study, because it's representing the human malaria in human, I think.
Yeah. Okay, thank you, Dr. Elsa.
Thank you.
Thank you, Mbak Asri. Yeah, thank you, Andita. So everyone, we approaching at the end of these sessions. Yeah, thank you very much for your presentation, Andita, Mesya, and also Wihda. Yeah, for sharing the study, which is interesting. Yeah, we learned a lot from this session about the potential of malaria target, potential drug, and then the potential drug candidate, and also attempt to develop malaria parasites model that resistant to artemisinin. So thank you very much, and I want to close the session, and remember that tomorrow there is still interesting topic to follow. Yeah. So thank you very much, and the judge, Dr. Henry, Dr. Asri, thank you very much also, and have a nice day for everyone. Bye.
Thank you, Dr. Elsa. Thank you, presenters.
Thank you, everyone.
Terima kasih.
Do we stay in the same room for the next? Sorry.
We could leave the breakout room. So this is the end
Okay.
of today's session, so we can leave the breakout room and
Oh,
do not
okay,
have to
sorry.
back to the gate.
Okay, sorry. Thank you.
Thank you all judges and all presenters. Have a great day.
Have a great day.